Dr. Wolchok, we'll start with you because we made you come up last. When you've been in this game or this field for a while, when a patient sees you for immunotherapy, what are the common questions they ask you when they first come to see you in the clinic?
So first of all, Chuck, thank you very much for leading this discussion, and thanks to all of you for coming out today. It's really a great opportunity for us to exchange information. I just want to at the outset state that I receive research funding and also am a consultant for all of the companies that are currently marketing immunotherapy medicines. And I think it's important for that transparency. I also think that WD-40 should be a new sponsor of this meeting next year. I'm happy to donate. To be serious, though, when a patient comes to us to talk about immunotherapy for melanoma, it's one of two major approaches to treating that disease that transformed the treatment of the disease. And actually, that along with BRAF-targeted therapy were the first interventions ever to prolong survival in a cancer where the median survival used to be about 6 or 7 months and now is greater than 3 years and counting. So we see the great impact of this. However, at the very beginning, it's important to stress, first of all, some of the points that you made, Chuck, that not everybody responds. Even in the best case, the response rate is 60%, which is still, it's hopeful, but we're not going to be ready to retire until it's 100%. And in addition to that, there are side effects that accompany immunotherapy, and those side effects are actually unlike the side effects that usually accompany other kinds of anti-cancer therapy. Many of us are familiar with the hair loss, nausea and vomiting, and decrease in blood counts that can accompany chemotherapy or radiation therapy. But immunotherapy, as you alluded to, Chuck, has a different list of side effects. They resemble the side effects of autoimmune disease. We know that we can dampen these side effects, but we also know that if we don't detect the side effects early, that people can die, and people have died from the side effects of immunotherapy. So we learned that good communication actually is critical, and that starts when we first introduce the idea of immunotherapy, that anything new that you or a family member is feeling is interesting to us. Some of these are not usual topics of dinner time conversation, like, hey, guess what? I had 15 episodes of diarrhea today. But no one is more interested in my patient's bowel habits than me, because we know that if a patient does develop diarrhea, which is a not infrequent complication from immunotherapy, the sooner we know about it, the easier we can turn it around. We also know that if we don't treat it with mechanism-based therapy, things like prednisone, which dampen the immune response, if we instead use common things like drugs that slow down the motility of our gastrointestinal tract, and I'll just pick on things like Imodium, right? That doesn't get to the root of the problem. We need to take the foot off the brake, or the foot off the gas, and really intervene in a meaningful way. And the sooner we know about this, the better. So at the very beginning, we introduce expectations in terms of the likelihood of response, and we also introduce the concept of the importance of good communication. I know that people often complain that their doctors are not available enough to them, that they seem busy and they don't get enough time, and we will try as hard as we can. We really need to know from you, and once someone calls and says, hey, I have a side effect, we then ask our nursing colleagues to follow up on a regular basis. We are available 24 hours a day because we recognize that early intervention is important.
That was a wonderful introduction to those side effects. I totally appreciate it. And speaking of diarrhea and colorectal cancer, Dr. Manji, you are an expert actually in colorectal cancer. And what I've heard actually is that there is maybe a biomarker that immunotherapy doesn't always work in colon cancer, but maybe there are some patients that it works really well in. Could you tell us a little bit about that, please?
Thanks, Chuck. That's a fantastic question. Like we suggested, that Chuck alluded to before, that not all patients respond to immunotherapy, And there are these predictive biomarkers that we need to understand. And what Dr. Drake is alluding to is microsatellite instability. And that is also what initially was introduced, that this is a fantastic biomarker because it is a biomarker that predicts response to therapy regardless of your histology, regardless of the type of cancer that you have. So if you have microsatellite instability, whether you have pancreas cancer, or whether you have colon cancer or gastric cancer, that immunotherapy sort of becomes a little bit frontline. So this is sort of personalized medicine that you should be looking for and should be asking your physician to sort of investigate your tumor. So in colon cancer, microsatellite instability basically gives us an indication that immunotherapy is what is important for your tumor to be treated. And going to the side effects, I mean, we do the same thing. It is critical. Sometimes many patients that are new to cancer, that are new to treatment, they say, it'll pass, the diarrhea will pass. Or I don't want to bother somebody, it's Saturday. But this is our job. This is why we are physicians. This is why we do clinical trials, and we take the responsibility very seriously. So as some of my patients over here will know, unfortunately, or fortunately for them, unfortunately for my wife, they have my cell phone. Okay, so if they're not able to get through to somebody, regardless of the time, you need to get help. And it's more important when you get admitted to a local hospital. They're not familiar with the clinical trial that you're on. And it's impossible for them to really understand what side effects to really look out for. And that's where you contact us. You tell them to contact us, and we can guide what needs to be done. So that's coming back to the side effect. But in microsatellite instability, it's not the percentage of patients with different histologies, with different types of cancer. The frequency of patients that have microsatellite instability also changes.
So I'm sorry to interrupt, but you keep using this big word, and you know I'm not that smart. We work together, right? So microsatellite instability, like what exactly does that mean? It sounds bad, but you're making it sound good.
Right. So microsatellite instability is basically what happens is that as a cell divides, imagine that you're given a flyer for CRI. And you're like, okay, we're going to make 1,000 copies of the pamphlets and we're going to give it to everybody. And whoever's the person who's on the printing shop is basically, who is not very paying attention to the job. So every time a copy is made, the second copy of the paper is used to make another copy. If you don't catch the mistakes early, you're going to start having a lot of mistakes. By the time you get to your 1,000th copy, it's hardly going to look like somebody who, of the original copy. So microsatellite instability is the checkpoint or the quality control of the DNA. So whenever a cell makes a copy, it checks to make sure that there are no mistakes. And when you have that error in that machinery, more and more mistakes are made. And that's when the immune system can recognize it easier that cell has a lot of mistakes, has a building where the doors are where the windows are supposed to be. So if you're driving by, you're like, this construction guy, something is wrong here. And you're going to investigate. And that's what happens with these tumors. So with microsatellite instability, there's so many mistakes on the tumor cell that it's easier for the immune cell to recognize that tumor and attack it.
So Dr. Diefenbach, I've also heard recently that there's some kind of blood cancer, like one particular one in which the immune system, these kind of drugs work spectacularly well. Maybe you could tell us a little bit about that?
Absolutely.
Some kind of lymphoma or something?
Yes, so it is so thank you, Chuck, for that very excellent question. I just want to add, for my disclosures, I do consulting and advisory work and receive research funding from pretty much every company that's developing immunology in the hematology space. So those are my disclosures. And I also want to thank all of you for coming out on a Saturday and listening to us so intently, and to Chuck for leading our panel, and to the CRI for inviting us. So I really am a lymphoma specialist, and the interesting thing is that lymphoma is one word, but it actually encompasses about 70 different cancers. And of those 70 cancers, each of them has a really unique and individual biology. Now, of those 70, at least 50 of them are probably T-cell lymphomas, which are very rare, but still there are about 20 or 25 B-cell lymphoma subtypes. And what Chuck is alluding to is that there's a particular type of lymphoma called Hodgkin lymphoma, which is really not like any other type of lymphoma, and it's not like any other type of cancer. So to understand why immunotherapy works so well in Hodgkin lymphoma, we have to take a step back to Hodgkin lymphoma biology, but I think doing that really tells us how we can unlock immunotherapy for other types of lymphoma and other types of cancer. And that's really to start by looking at the cells. So colon cancer grows in sheets of cancer cells. Melanoma, if you look at it under a microscope, you see sheets of melanocytes or melanoma cancer cells, and they all look the same, and each one looks just like the other. In Hodgkin lymphoma, what you see is one or two tumor cells, which are called Hodgkin-Reed-Sternberg cells, that are these big cells that look very characteristic. They have two eyes that look like owl eyes, which are their organelles. So you can see them under a microscope. But what you see one Reed-Sternberg cell, you do not see another one. Because in one field of looking at the tumor, you might see 1 or 0 or 2. And around these Reed-Sternberg cells are the cells of the patient's own immune system. So the T cells, the B cells, macrophages, which are the scavenger cells of your immune system, dendritic cells, which are the cells that gobble up parts of cells and then present antigen to activate the immune system. They're all sitting around these Reed-Sternberg cells. But are they eliminating the Reed-Sternberg cells? Are they doing what they're supposed to do? No. They're actually sending chemokine signals to the Reed-Sternberg cells that help them both evade immune detection and grow. So in looking at Hodgkin lymphoma back before there was any immunotherapy in the Hodgkin space, I and others asked the question of why is the immune system tolerating the Reed-Sternberg cells and not waking up and eliminating them since there are so few tumor cells? So we took the immunotherapy that was then in melanoma and in the solid tumor space and said, if we take a PD-1 inhibitor like nivolumab and give it to these T cells that are in the Hodgkin space, will it wake up the T cells and help them fight the tumor? And I developed a trial that is now a national trial where we took ipilimumab, the CTLA-4 inhibitor that Chuck described, and nivolumab, and gave it in combination with a targeted drug to target the tumor cells to try to wake up the tumor cells in the Hodgkin space and turn them against the cancer. And I and others have shown that nivolumab is exquisitely effective for Hodgkin lymphoma.
Like how effective is exquisitely? Like 50%? Like I got 30. I was pretty happy with that.
So with all the chemotherapy drugs in Hodgkin's, the overall response rate is about 30% for relapsed Hodgkin lymphoma. A drug came along called brentuximab vedotin, and that had about a 65% response rate and a progression-free survival, so the amount of time that you didn't progress, of about 5.6 months. Immunotherapy, nivolumab, came to the Hodgkin space and had a response rate in patients with heavily relapsed disease of about 80%. That's overall response. But with just the PD-1 inhibitor, the complete response was only 9 to 15%. Now we know that many of those with stable disease or a PR will continue to do very well. But when we combined nivolumab with a targeted agent, we had a complete response rate of 64%.
Wow, now you're talking.
And we actually are now looking at the combination of both the CTLA-4 drug to take the foot off the brake and the nivolumab to wake up the exhausted T cells with brentuximab, and that data we'll be presenting this year at the ASH meeting. That's the first time that double immunotherapy with a targeted agent has been shown in Hodgkin's, and I can't tell you what it shows, but we're very excited.
So combination immunotherapy in Hodgkin's sounds very exciting, actually. So, combination immunotherapy in melanoma, in the trials we give, 4 doses or something. Does everybody need all that? How do we decide how much combination immunotherapy? What do you think about that?
That is a great question, Chuck. I also want to congratulate everyone because I think you're now all ready for a master's degree after all that information. This is like the education building, so
it is the education.
I like to just pause for a moment and say, what did we just learn? Before we talk about combinations, from my colleagues over here, how does someone who doesn't have Hodgkin lymphoma or colorectal cancer that has a lot of genetic errors in it, how does a person put all this together and understand who's responding to immunotherapy? Because that may tell us a little bit about why some people need one drug, why some people need two drugs, why these drugs don't seem to work for some cancers. So in sort of like my own Cliff Note version of the master classes that were just taught, what I take away from this is that there are some cancers that the immune system is interested in. And those cancers have a lot of genetic mutations, a lot of errors in the copying that was made. Or they have an inherent quality, like the lymphomas, that make the immune system interested. Now, the genetic error part is interesting because there are some solid tumors that are very, very genetically mutated. Melanoma is sort of the quote-unquote poster child for this because it occurs in many people as a result of exposure to sunlight, which causes an accumulation of genetic damage in the skin. So there's a lot of genetic noise in melanoma, which makes it look different than the normal skin cells before they were exposed to sunlight. And so the immune system sees the doors where the windows should be. I like that analogy. I'm going to use that. And it says, aha, this is something abnormal. It's something different. Maybe it's something that looks like a virus or a bacteria, like Dr. Coley did 120 years ago. So I'm going to respond to that. The other diseases where this happens are in bladder cancer and lung cancer and some other upper airway and upper aerodigestive cancers, meaning upper gastrointestinal cancers, because of tobacco exposure. And also in kidney cancer, where there are differing reasons for genetic damage, but that is one reason why some cancers are amenable to the use of immunotherapy, because they look different than the normal cell that they came from as a result of genetic damage, either because it accumulates as a result of a genetic syndrome or because of exposure to some type of carcinogen. So with that said, we know that some people with melanoma can have great responses to one immunotherapy drug, and some people need two. We are not sure how to distinguish those two possibilities yet. One of my colleagues, Michael Postow, is doing a trial to try to dig into this a little bit more deeply. But this is an important question because, as you heard from Chuck, two of these medicines have many more side effects than one does. The side effects are not different, they're just more common. And so why would we expose someone to more side effects if we could just get away with one medicine? So that's an important question that is still open for us to answer. And I don't have an answer for you yet, except that we are embarking upon studies that are looking at earlier time points after maybe one or two doses to say, aha, the person's tumors are shrinking, maybe we should back off now rather than aiming, so the more is better. Let's pile on more medicines and give it 4 times because that's what the clinical trial said. We're not sure that we need to do that. So we are actively engaged in that to try to find the right balance between risk and benefit. And that's obviously very, very important.
And that sounds like a very intelligent trial. In kidney cancer, we're doing a more empirical or guesswork trial. What we do there is just give the anti-PD-1 first, give it a little shot, give peace a chance. If it works, we're happy. If it doesn't work, then we add on the other drugs. So trials to try to figure out who needs one and who needs the other. But not all cancers respond so well. And one of the cancers that's really been a challenge is pancreatic cancer. Is there any exciting new research in pancreatic cancer that can help the immune system work in pancreatic cancer?
Right. So pancreas cancer is my research specialty. And when Dr. Drake showed those T cells that were within the tumor, where he described hot tumors or inflamed tumors, and then there were these cold tumors where there's not a whole lot of T cells in them. And the way that I describe it again to patients is also about, it's not just about the building, it's about where the building is, right? It's about the neighborhood. So imagine pancreas cancer is a gated community. Doesn't allow the T cells to come in, right? So you can't just think about, having doors where windows should be, but if it's a gated community where the T cells just can't come in and even recognize, that tumor will go unchecked, right? So you have to think about, how to get these T cells activated, how to get the T cells in. And in pancreas cancer, that's the problem. One of the problems is that there is not just a gated community. Unfortunately, the supportive staff in the neighborhood is actually cooperating with the T cells to not allow the T cells to come in and not allow to be recognized. So, again, with the analogy, not in the gated community, you unfortunately have a house with a meth lab. So what they're going to do is, so if you were to, I'm not supporting this, but if you were to want to set up a lab, you're not going to have, what would you do? You would have a nice house, you would have, swings, you would have sort of a sandbox, you would have a pool, you would have all these, you would have a camouflage. And you would sort of cooperate with the neighbors and the environment to keep it quiet. And that's what pancreas cancer is. So
what can we do about it?
So what can we do about it? So that's what we're I have a lab over here where we have a mouse model of pancreas cancer. We have taken mutations that we know are important in the pancreas cancer to develop. They specifically are expressed in the pancreas tumors. These mice do develop pancreas cancer, and then these tumors actually then spread to the liver and behave very much like the pancreas tumor in patients. And what we do is we treat them with immunotherapy, but then we treat them with different combinations where we take the gated community. So, for example, if there is a gate, we try to tweak and add agents that will affect the gate. Then there are also supporting cells that cooperate with these tumor cells so that they can grow. So we also go ahead and target those cooperating cells. And then we test multiple different combinations to see which combinations lead to tumor shrinkage and the mice to live longer. And then we propose a clinical trial. So there are multiple, unfortunately, in monotherapy, unlike melanoma, immunotherapy in most pancreas cancer hasn't worked. But in about 2% or fewer patients of pancreas cancer, they do have that signal where doors are placed instead of windows, so microsatellite instability. With their pancreas cancer patients will respond to immunotherapy. So there's a clinical trial ongoing at Columbia in multiple different places where what we're doing is we're treating patients with not just immunotherapy, but multiple different combinations that are targeting the gate in the gated community. They're targeting with the cooperating cells that are not allowing T cells to be effective, and that's how we're going.
Any luck?
Can't talk about the results yet, but there is hope.
That's great. When we talked about the biomarkers, we talked about things in the tumor. Is there PD-L1? Are there immune cells there? But the body is complicated, and one of the things that the body is notable for is there's probably more bacterial DNA in each of our bodies than there is our DNA. So one idea as a biomarker is all those bacteria are kind of affecting your immune system, making you maybe more likely to respond to immunotherapy. I've heard that there's some research in this field as well.
So I think what you're alluding to is something called the microbiome, which is really all the bacteria and virus that live in your gut and sort of form a neighborhood in your gut that interacts with the immune system. But before I get to that point, I want to say that I think what's really interesting is trying to understand not only the immune system near the tumor, but how the interaction of the immune system with the tumor interacts and affects your immune system away from the tumor. So that's sort of like think globally, think locally, act globally. And basically the question is there an impact on the global immune system from having a tumor? Is there an impact on your T cells that are not in the neighborhood of the meth house or anywhere else, but somewhere else? And so one of the questions that we ask is really how does having the tumor affect your T cells in general, not just your T cells around the tumor? And how does your bacteria and viruses that live in your gut potentially affect your immune system to allow a tumor to grow, to allow this bad house to be put in your neighborhood or not. In Hodgkin lymphoma, which is where I've done most of my research, what we've actually seen is that patients who get immunotherapy tend to not have the same degree of toxicities as these solid tumor patients do. So they get a little bit of diarrhea, they get a little bit of rash, but it's actually been very surprising that we've given the same doses of immunotherapy in the heme space as our solid tumor friends have done, and we've seen much less toxicity.
That's good.
So,
you should be happy.
Why is that? So we looked in the T cells that are not at the tumor, but the T cells in Hodgkin's patients that are just floating around. And it looks like in many of them, the T cells are all exhausted, not just the T cells at the tumor site. So does that mean perhaps that if your T cells are quite exhausted, they're tired all over, you're going to have less toxicity? Maybe. When we look at the bacteria and viruses that live in the gut, we're not really looking because we're interested in, do you need an antibiotic to clean out your gut? Because your gut is actually never clean. The bacteria and viruses that are in your gut have been with you since the minute you're born, and they're the way they are because of where you grew up, who your parents are, what you ate, and what antibiotics you got, especially the antibiotics you get when you're young. But what we're really interested in is, do patients with lymphoma, and remember that there are 70 kinds, is the aggressiveness of their lymphoma at all related to what kind of bacteria and viruses are in their gut? And is the response to therapy at all related to what kind of bacteria and viruses are in your gut? Now that's not research that's very far along in heme at all, and in solid tumors a little bit, but it looks like there are some strains of bacteria and some having more or less of certain bacteria that may impact the actual behavior of your tumor. If this is really the case, this opens up a whole new avenue of being able to target the immune system. Maybe you take a couple of probiotics and you can get some good bacteria and change your immune system. This is not ready for prime time yet. Chuck is asking me about stuff that's very early in development, but it's an exciting development of the field. Certainly not only and very early in the heme space, it's in melanoma and some other solid tumors, it's actually much more developed, and there have been some very interesting discoveries. And I would say really all this is saying is that the immune system is not just the interaction of the tumor and the T cells. It's really the global immune system in your whole body and its interaction with the bacteria and viruses that live in your body in the context of having a tumor that's going to unlock our ability to help the immune system target the tumor better.
And Dr. Wolchok, as we heard, this has been, there have been some studies of this microbiome stuff in melanoma. Do you have any comments on that?
There have been studies, and I think it's still quite early. We still don't know whether there's something that you can ingest, whether there's a particular probiotic or prebiotic that you can eat that will affect your likelihood of response. But in both animal models and in retrospective studies of people, there does seem to be a relationship between the particular bacteria that are in your intestines, in the bacteria in your intestines, and the likelihood of response to immunotherapy. It's not 0% or 100%. I think what we're learning is that it's one thing that modifies the likelihood of response. But bacteria, and we've heard a lot about bacteria today. We started learning about bacteria from Dr. Coley. Now we're hearing about how bacteria may modify our body's immune system. But I actually think that it's important to take a moment to think about bacteria and viruses, because these pathogens, these things that we think are dirty and dangerous, actually are being used now as a way to remedy something that's been referred to before as the cold tumor problem. Right?
Yeah, tell us about that.
Yeah, you referred to it.
I did. I had a picture too.
Right, so the cold tumor is one that has not attracted the attention of the immune system yet. And it doesn't have a lot of genetic damage. It's not very interesting like some lymphomas are. So, these are a lot of very common solid tumors. I was talking with a gentleman earlier whose son is being treated for colorectal cancer, not the kind that actually has all that genetic damage. So what can we do to make a very common cancer like colorectal cancer more interesting to the immune system? Well, there are a few things that are being tried, and this is early clinical research. One is to inject it with engineered viruses. Take one metastasis, inject it with a virus. These are called oncolytic viruses, cancer-killing viruses. And that does two things. It kills some tumor cells, a few, but it also, for a short time, makes the cancer look like it's infected with a virus, and that might attract the attention of the immune system to the cancer. And if you follow that by one of these checkpoint blocking drugs, one of these step on the gas or release the brake drugs, you then might be able to sort of start the engine of the immune system. The other thing that we can do, unrelated to bacteria viruses, is to say, you know what, we need to put something into the blood of these patients or redirect the T cells in the blood of these patients to make them all of a sudden interested in these otherwise cold tumors. And this is common in some breast cancers, in colon cancer, in other common cancers. How do we redirect the immune system? And that's really, there are some clinical trials of what are the CAR T cells, which I'm sure you'll hear about from Dr. Diefenbach, because they're being used in blood cancers, and you heard about it in terms of Emily Whitehead's, fantastic result. But in solid tumors, the targets are more difficult to find, and we're looking at ways not necessarily of expanding cells outside the body, but of redirecting them while they're still inside the body, with what are called bispecific or two-headed proteins to kind of...
Oh, that sounds interesting. Tell us about that.
Yeah, so it's...
Bispecific.
Yeah, so it's basically a protein that's been engineered to have a piece of Velcro on one side of it that attaches to the cancer cell, and another piece of Velcro on the other side that grabs any random T cell floating by and makes it stick to the cancer cell and kill it. So it doesn't so much matter what that T cell or that immune cell was originally programmed to recognize, but because of this two-headed Velcro molecule, we're seeing some activity in diseases like colon cancer.
Wow, that's fascinating, actually. Going back to colon cancer, we had a little show of hands before. Who heard of immunotherapy? Who here has heard of chemotherapy? Very good, actually. Dr. Manji teaches patients with colorectal cancer, and he gives in his practice some immunotherapy and some chemotherapy. I wonder if you could contrast for the folks here the side effects and what happens to patients who get immunotherapy versus chemotherapy.
Right. So before I answer that, I just wanted to talk about the CAR T cells and these two-headed molecules. So, there is actually now even second generation, these two molecules that are coming, that are sort of being tested now. There's also interleukin cytokines. Cytokines are chemicals that inflame, that make a cold environment sort of hot, sort of bring attention to the immune system so that they can sort of, the immune system can recognize that area. So now there are actually, these chemicals are being bound to these targeted molecules, these so that they can bind particularly to the flags that these tumor cells express and make it more inflamed. There are also some molecules that are going to go attack the neighbors, the collaborating neighbors that are not so good. So a lot of exciting targeted molecules that are being tested right now, at centers like Sloan and over here. There's also a CAR T cell, and then remember the CAR T cell technologies where you take the patient's T cells, you bring them out, and you re-engineer them so that they can detect the tumor. So that's a trial that's going to start soon at Columbia also. So going back to Dr. Drake's question regarding the chemotherapy side effects. So with colon cancer, usually we use combination chemotherapy. I don't know how many of you have heard of 5-FU pumps. You go home with a pump, you come back with a pump, or you can take the oral chemotherapy equivalent. The side effects are we have to always worry about the blood counts. It puts you at increased risk for infection, fatigue, increased risk of bleeding. There's also sometimes mucositis, which is sort of ulcers in the mouth. You have difficulty eating. And the other chemotherapy, one of the chemotherapies can cause profound diarrhea that can really get somebody into trouble. And then another chemotherapy, which is really, it's limiting, very, very effective, called oxaliplatinum, which causes numbness and tingling. And the numbness and tingling, after 6-8 cycles is really debilitating to the point where we have to reduce the doses, or we have to basically eliminate it altogether.
So it's all bad. Numbness, tingling, nausea, diarrhea. How about for immunotherapy?
So immunotherapy, the side effects are very different, okay? Generally milder, but some of the side effects can be serious, as alluded to by Dr. Drake. So you can have, every time I have somebody on immunotherapy, I'll always ask fatigue, because there could be profound fatigue where the immunotherapy drugs can affect part of the brain that make hormones for us to be active. So, for example, if you go in and you have a really important meeting and you're trying to sell your project, you're going to have, endorphins, steroids. Basically, you're going to be pumped up. How they say, get your game face on? Well, that is basically gone, right? You're extremely fatigued. You cannot get out of bed. So that is an alarming sign. Also can affect the lungs. So if you're having shortness of breath, persistent cough. So these are all autoimmune type side effects where the immune system, instead of recognizing the tumor, is starting to recognize self. And diarrhea can also be profound, but not as common depending on the drug that you're using and the combination that you're using.
I mean, I would agree. I think about 15% is what we tell people with kidney cancer for one drug. So those were my questions of the panel. We're now going to turn it over to you, the studio audience, for questions for the panel. Brian will moderate that part, and you could even ask me a question. We have folks, I think, running with microphones. Is this correct?
Yes.
And so if you have a question,
and we'll just ask, since we can't get to the middle, if you've got a question in the middle, that the folks on the ends help pass the microphone down as quickly as possible. Before we begin, though, I just want to remind you all to be sure to set your DVRs to record next week's episode of Breaking Bad. All right, and also just another note, this is an open forum. Everyone's got lots of different questions. I'm sure everyone is concerned about their particular type of cancer, but in this forum, I would ask you, as you think about your questions, to frame them in a way that they would be of interest to other people in the audience. Later on today, you'll have an opportunity with that one-on-one interaction or in their smaller breakout sessions to get a little more in-depth into your particular type of cancer. So does anyone have a question for the panelists? We have, all right, one at the back. Also, I'm sorry to interrupt you. We have folks watching via live stream also, and I'd like to remind them that they're also able to submit questions. We are monitoring them, and we'll be sure to pass them on. Okay.
First, thank you for hosting this. It's been great. And the question I have is, in my situation, my wife has colorectal cancer, but the general question I think is, when your microsatellites, they will have mutations like KRAS, what do you recommend? Because when you read literature, it seems like for many cancers, KRAS mutant and MSS go together, and the immunotherapies, at least from what I read, generally don't sound like they work. So any suggestions or how we should think about that?
So in general, I think that MSS patients have a lower response rate, much lower response rate, so immunotherapy would not really be front and center. There was a clinical trial that was done with a targeted therapy in combination with immunotherapy. Initially, the results were a little bit promising, where it suggested that you could turn, regardless of your MSI status, you could sort of make a more inflamed tumor, and immunotherapy would work. But unfortunately, there was a large randomized study that was done where immunotherapy, unfortunately, with that combination, was not really shown to be effective. And those results are going to be, coming out at ESMO, I think next month. So right now, I would say that chemotherapy would be upfront in those type of patients.
But we would both add, after standard therapy, all these drugs that are now approved came through clinical trials, actually. And so if standard therapy is not working, actually, even for challenging tumors, there are many, many, many combination immunotherapy trials that are worth thinking about.
I would also add that the particular trial that Gulam was referring to used a medicine that blocks a very important protein called MEK, which is just downstream of RAS. It's sort of in that circuit that connects the outside of the cell to the nucleus of the cell to tell it to divide. And RAS is sort of like KRAS, which is the most mutated of all driving cancer genes, is the holy grail in cancer research, to find medicines that block RAS very well. We're not there yet, but we're working on it. So we're settling for the moment for the downstream molecule MEK. And what Gulam said is absolutely correct, that we had a lot of high expectations about combining MEK inhibitors with some of these immunotherapies. I have the privilege of being part of a Stand Up to Cancer dream team that's looking at this problem in KRAS mutant lung cancer, which is the largest segment of non-small cell lung cancer. What we have found in some very early work is that when the medicines are given as they were in that clinical trial, meaning combined together all the time, constantly, that's not so good. But if you give intermittent bursts of the MEK inhibitor, so high doses not very often, there may be some advantage to that combination. So we're hoping to revisit this concept in clinical trials using different schedules and different doses, and to see whether we can find some activity, because there's still much more to learn.
Right, and then in that early trial, there was some suggestion that you may have some activity where the T cells may be coming in with just MEK inhibition. So even though the trial was not positive, we can learn a lot from it, and that there might also be a biomarker where you can selectively pick a certain proportion that would be more applicable for that combination. And the issue is with the KRAS mutated, 95% of pancreas cancer patients have KRAS mutation. And that is what is the challenge. So if we can, target KRAS, and some of the clinical trials that are actually ongoing are doing that with RNA targeted therapy, and then combining it with different therapies to see whether that's effective.
Okay, we have a question up here. Yes.
Hi, doctors and doctors. First of all, this is wonderful. I've had cancer for over 5 years now, and I assumed as it went on, and I educated myself more and more, it get easier and easier. And with this immunotherapy and T-cell therapy, it's like you're watching a sci-fi channel. It's just amazing how high-tech it is. So my question is, Dr. Manji, you were talking about re-engineering cells, taking them out of the body and re-engineering them. And that's something I still have a tough time understanding. Is it a chemical re-engineering, or is it some kind of mechanical?
Excellent.
How does that happen?
So let me try to answer that one, because it's mostly in the space of blood cancers where they're doing this. So I think Chuck alluded to it as well. So basically, they collect your T cells, which are your immune cells. They take them out of your blood. Then they actually, in the dish, re-engineer your T cells. So they basically they genetically change your T cell. So they engineer your T cell with what we call a chimeric antigen. And what that is a fancy word for basically giving your they take some tumor cells and your T cells, and they engineer your T cells to be able to recognize the tumor cells that are in your body, and to activate the immune system to come and kill those tumor cells. So they basically take a T cell that's not doing its job very well, genetically engineer it to do its job better, and then they put it back in you. Now, That's a specific type of immunotherapy that is not really like these checkpoint inhibitors that you just get in an IV. It's a much more high-tech version of immunotherapy that comes a little closer to the old-fashioned stem cell transplant, because you then have to get these T cells back. In order to get these T cells back, they have to give you medicines to get your other T cells lower so that you will accept these T cells. And there are toxicities related to the whole process of getting the CAR T cells back that are unique toxicities in immunotherapy. Particularly, the two that are most common in the CAR T cells are neurologic side effects, so seizures, cognitive changes in your thinking that go away most of the time and get all better, but not all of the time, and changes in your blood pressure, so big drops in your blood pressure where your body almost acts like it is in the threat of a big infection. And it's doing that because when your immune system is activated to the high degree that these CAR T cells do, it causes all the signaling molecules that your immune system uses, what we call chemokines and cytokines, to basically just burst out like a hose that all of a sudden all the water is splashing out. And when it splashes to that degree, it lowers your blood pressure. It's very similar to if your body's fighting a bad infection. So you really need sometimes to get these medicines in the ICU with support for your blood pressure and checks for your brain. So these are not trivial medicines. They're complicated medicines. They need to be given in a specialized center, but they have really changed the lives of people with aggressive lymphomas, and patients who had no cure on the horizon have been cured with aggressive lymphomas and leukemias because of these cells. But they don't work in everybody, and they have a lot of toxicity. So what we now have to figure out is how to make these drugs safer, easier to give, and to work better in a larger number of patients.
So just to get back to you, is basically you're taking the T cell that's not functional and that's not recognizing tumor, and you're changing the Velcro. Okay? You're reprogramming the Velcro so that it recognizes the tumor. And you know, with the clinical trial that's going to be starting at Columbia and it's already in Houston, is that you're putting a part of a DNA that makes these proteins into the T cells. So you take the T cells out, you introduce something that changes the Velcro, but it also has a payload that you can engineer in. So the exciting part that I think is that you can make that T cells also make things that also affect simultaneously the neighborhood that they're going to be in. Okay, so if the neighborhood's going to have a gated community, you can not only engineer the T cells to go to that neighborhood where the tumor is, but that T cell will also make a chemical that will open the gate. Make sense? So you can engineer that part. And this is science fiction right now, but we're going to be testing that in the lab.
We are in the presence of a master of analogy over here. All right, we have a question here.
Thank you all for being here. This is great information so far. Some background. My mother has stage 4 pancreatic cancer. She's been on 2 clinical trials that she's progressed on, one being a combination immunotherapy clinical trial. My question is more broad about trial design in general. I think one of the things just being on 2 trials was the ramp-up period. You had to be without any medicine for 30 days, and you have to go through the additional scans. You have scans in another hospital, have to go through with the hospital that's going to be doing the clinical trial. And a lot of that's wasted time, just, the patient is getting worse, and some people don't even make it on the trial, even though they could have started on day 1. There's a couple of trials right now being done, one at Sloan Kettering and one going to be done in multi-institutes for pancreatic cancer called Precision Promise. But I think there's also one for pancreatic cancer right now at Sloan Kettering doing, basically being able to move within cohorts. So if you progress on a combination immunotherapy, then just move on to the next one, the next one, and the next one. Within the same facility, so you don't have to get additional testing, you don't have to move from doctor to doctor. I'm just wondering, in general, is that where we're going to be going in terms of doctors and MDs pushing for that new type of trial design, especially in highly lethal cancers?
Right, so I think that if it's okay if I can take that. So I think that you're discussing the Roche study, which is a Morpheus study in pancreas, and many other different pharmaceutical companies are using the same design. So just so for everybody else, usually traditionally the clinical trials were designed such that you would just, test one combination. You would get on that clinical trial. If the clinical trial worked, you would continue. If the clinical trial did not work, you would come off that study, and then you would go through the whole process all over again. So in pancreas cancer, which unfortunately is an aggressive tumor, most of the time you need to get things done quickly and get started quickly. This new design allows you to randomize. There are multiple cohorts, so there are multiple different arms. So there's not just two arms. You get randomized to one, and then you're followed. If you respond, you continue on that study on that arm. But if you do not, very quickly you can then progress on to the subsequent arms, and that makes to me that makes sense. It's efficient trial design, and now it's being done in many different histologies for what you mentioned as far as the time. The question regarding the CAT scans and all of those things that need to be done, and why there is a washout period where you have to wait. The reason for the washout period is that if you are on a prior clinical trial or if you are on chemotherapy, sometimes the side effects can manifest a little bit later. If you are testing an early phase clinical trial in which the focus is really also to determine safety, that can be a problem where you might not accurately be able to distinguish whether the safety issue is because of the new combination or the combination that you were treated previously. So that's the reason why. The reason to get a CAT scan quickly done is because you need to have an accurate baseline with scans to make sure that you can determine that.
So I mean, honestly, and Brian will do a great job talking about this later, but all trials are a balance between trying to help patients with new drugs and new combinations of drugs and doing really an experiment to test whether that works, right? And some of the things that you're talking about are part of the experiment to get a baseline, and other things are the parts that you're talking about, right? Because we want to try that drug right away to see if it helps, actually. That's a balance we all work with every day, and it is a challenge. We all work together to try to make trials as easy to get on as possible, but we always need them to also be safe for the patients, right? And also to be able to answer the question, right? Does the drug work or not? And so you point out a very important concept, which we're all working on, frankly.
And I'd also say that hearing things like these comments from you is very important to us, because when we are all part of many organizations that bind the oncology community together. One of those is the American Society for Clinical Oncology, or ASCO. ASCO has a task force right now which is looking at eligibility criteria for clinical trials to try to make these more accessible and inclusive. So to give you an example, some clinical trials exclude people if they had a different cancer that was cured a short time ago. And that's, frankly, in my opinion, unfair. So we are now actively working across the community with many organizations and the FDA, actually, to really enact clinical trial reform so that we can make this eligibility process much more friendly to patients and their families.
And I think, part of this is that although trials are supposed to test drugs in a representative sample of patients, most of the patients who enroll in trials is just a small cross-section of who patients with cancer really are. Patients who are over 65 have very little representation in patients who go on to clinical trials, because the eligibility of trials is written in such a way that it's very hard for anyone except the fittest elderly patient to go on to a clinical trial. Patients in rural communities are on clinical trials much less than patients who are in urban communities, because they don't have access to clinical trials. And these are real issues about equitability and trying to have the research we're doing represent a true cross-section of people who have the disease. And these are things that Jedd and myself and others are working at trying to revise. So I don't think trials that say you can only have had 2 times therapy but not 4 times therapy are really doing a very good service to patients in trying to get the drugs to the patients. these basket trials that you're speaking of, where there are different options offered, and you can go from one option if it doesn't work to another one, is one of the ways that we're starting to design clinical trials with a little more thought to patients. So why should you have to have scan and then come off a trial and then have another scan 6 weeks later if it doesn't work? These are really important questions, and hearing them from you helps us design better trials that are more patient-focused.
So we have time for one more. Do we have one on this side of the room? Yes, okay.
Good morning. I'd like you to talk a little bit about the research that is currently being done on small cell neuroendocrine tumors of the uterus in particular.
Does anyone have an expertise in that?
Small cell neuroendocrine
Of the uterus.
of the uterus. I mean, I'm not an expert of that's a very specific question, but I think that if it's a small cell neuroendocrine tumor, you would treat it as a basket trial, which behaves like small cell. Not really sure if there are any specific trials designed for that particular.
No, so we have. So small cell tumors might have derangement in not the genes, but things that control the genes called epigenetic effects. So there are actually some very promising trials combining epigenetic drugs with either immunotherapy in small cell tumors, actually. That's one of the concepts, is that it's a different series of drugs, actually.
All right, well, unfortunately, that's all the time we have for the Q&A panel, so let's give our experts a round of applause.